Welcome everyone, my name's Richard MEYER. Today I'm going to talk about current research and opportunities for canola meal as a protein source in Australia. So looking at Australian production, normally we sit around 2 to 3 million tonnes per annum with 800,000 to 1 million tonnes processed domestically into meal. Of the canola that we export, about 2/3 goes to the European Union, and with the balance going to Southeast Asia and the subcontinent. So looking forward to canola production in 2021-22, we actually got very favourable conditions this year, and so there's been a 24% increase in plantings, and we're expecting to have an 11% increase in tonnage up from last year to 5 million tonnes, which is quite a fair way above our average of about between 2 and 3 million tonnes. And this is going to boost our exports as well. We're looking at a 15% increase in our exports to 4 million tonnes per annum. And it's actually a perfect storm for Australians right now. We're looking at a 41% higher price than the 10-year average due to tightening worldwide supplies and average climatic conditions in the Northern Hemisphere. So that's going to look at boosting Australia's exports by about 31% in terms of revenue. So this is where Australia sits as far as production compared to the rest of the players in the world. So obviously we're still one of the little players in the world, but yeah, we're boosting our production this year. Looking at the actual Australian canola crush, about 2/3 of our canola is solvent extracted, which leaves about 1/3 that's expeller and cold pressed. And there's also a diverse mixture of GM and non-GM lines of production. Obviously compared to solvent extracted meal, expeller meal has a much higher oil content of about 9 to 15%. This does have a couple of consequences. It means that there's higher energy available for our animal production. So for every 0.4% of oil content, there's an increase of about 1 megajoule of energy, which is beneficial to animals. But on the flip side of that, nutritionalists have to be careful that they don't go over that 5% level that starts causing problems for ruminants in terms of fibre digestion and depressed intakes. Now, if we look at Australian canola production as far as the Australian map goes, you can see that most of the production is centred around the southern states, southern more temperate zones, and over 50% is produced in Western Australia. The next map shows our processing area. So you can see a lot of our processors are located in the eastern states, and this is because they're close to our livestock, poultry, and also our centres of population. So a bit of a breakdown of canola use in Australia. Poultry and dairy obviously have the greatest share of our domestic canola meal that's produced, and you can also see our potential areas of expansion in beef and lamb and layer areas down in the bottom of the graph. So where does canola meal in Australia fit in price-wise compared to other comparative meals? Well, it's actually very, very competitive and in terms of cents per megajoule energy and also dollars per kilogram protein. So if we look at with or without or solvent extracted meal and expeller meal, you can see that they're very competitive compared to the other meals that are on offer. If we look at trends in canola meal R&D in Australia, we can see that there's a fair bit of innovation from our processors that are looking at value-adding products, especially in ruminant livestock areas, because there's been massive growth in prices in livestock areas over a number of years now. So that industry is booming. Also, ongoing research supporting dairy, feedlotting, and grass-fed livestock, and a lot of focus on nitrogen protein efficiency, obviously, as a protein meal, and increasing interest in innovative products to address increasing demand for human nutrition products. So now I thought I'd have a bit of a snapshot of some research that's going on in Australia. This is by no means exhaustive, but something I could do in this time. So first of all, I'm going to look at reviewing the project of the National Brassica Germplasm Improvement Program, or MBGIP as it's known, and potential improvements for canola content of meal. And that was headed up by Jamie AYTON from New South Wales DPI, and I worked on that myself as well. Next, I thought I'd have a look at two other research projects that were conducted at Charles Sturt University under Gay Krebs, with Rebecca Heim looking at a survey of protein fractions in locally produced meals and using some newer technology for rumen undegradable protein. And then I thought that we would have a look at Emma Lynch's project, which centres on researching feed and nitrogen efficiency in canola meal as a supplement to grass-fed pasture-based programs. So if we look at current breeding programs in Australia, they focus predominantly on yield and oil content, but they also look at disease resistance, drought and heat tolerance and frost tolerance, which was related to drought, and dual purpose crops. So there's been a fair, I guess, focus on those areas, with Australia being heavily in drought in 2017 to early 2020. Been a fair uptake of that. And obviously there's opportunities to increase the quality of the oil and the meal and the total crop value as well. So if I look at the first one of those research projects, which was headed by oil chemist Jamie AYTON from New South Wales Primary Industries, the aim of that program was to determine the genetic potential of a diverse set of canola germplasm for production, oil and meal quality traits. And if we look at the layout or design of that program, we looked at 91 genotypes against 4 controlled varieties across 6 field sites that represented low, medium and high rainfall zones. And they were spread right out across those that map that you had seen earlier, encompassing New South Wales, Western Australia, South Australia and Victoria. We did a lot of lab analysis on those particular varieties, but I'll only just focus on protein and oil content in today's, and energy content in today's talk. So if we look at oil content, this graph shows the 3 best performing varieties and the 3 poorest performing varieties, and we can see an obvious significant genotype effect. And I guess we note that the mean oil content in Australia has been between 41 and 47% over the last decade. So they're not outstanding performers there, but there is a significant genotype effect. The other thing that we have noticed as well is that increasing rainfall also contributes to higher oil content, as discussed by Meyler and Pratley and Pritchard as well. If we now look at protein content, and this is a fairly busy graph, but I'll work my way through it. So we've got our 6 sites represented by low rainfall, medium rainfall and high rainfall zones. And each one of these box plots represents the performance of each of those varieties in terms of protein content in that year at that site. So, and in addition to that, you can see the X that's in those box plots represents the performance of the control varieties. So you can see that there's many varieties that are performing better in terms of protein content than those control varieties. If you look at the top zone of that graph, I've also got total rainfall on there between April and November. And what we tend to see is the higher a relationship, where there's an inverse relationship, where the higher the rainfall, the lower the protein content. And you can see that site-related effect there. So there's some fairly significant environmental effects there from those zones. And I've also included a potential cost there as well, or premium for say 10%. So 10% of extra protein would be worth an extra $130 a tonne. Now, if we look at that in a bit of a different way, we've got the 3 best performing genotypes and the 3 poorer performing genotypes and a control there as well. So we can see a significant genotype effect and an opportunity to increase protein within the meal if we wanted to. The thing that strikes me about this graph as well is I guess the consistency of those higher performing protein types compared to the controls. We can see a much narrower spread of values within those particular varieties. And just to note that the Australian mean protein content observed is between 37 and 41%. So they're performing potentially a lot better, those varieties. If we have a look at that in a different way too, we've spread out the variation into genetic and environmental, I guess, types. And we can see that the genetic and the environmental influence is pretty much on par. So it does show the effect that the significant genetic potential for significant genetic improvement, especially compared to the controls. Just looking at a general trend that we're seeing, we've seen an inverse relationship between rainfall and protein content, which has been noted by these authors as well. But that was pretty obvious from this study. When we have a look at the correlation between oil content and protein, obviously we see a pretty strong negative relationship there. So oil is obviously the most valuable commodity that's gained out of canola versus protein meal, which is secondary. So what we don't want is to see high protein at the expense of a lower oil content. But what we see with these 4 varieties here is consistently high oil and protein content. So they're 4 varieties. They're amongst those varieties that are circled there on that graph. Now we look at animal production figures, protein versus energy content, and you can also see a strong correlation. This isn't really a surprise because we know that protein makes up a large part of the energy content of those feeds. But we can also see that correlation is really strong for fish, pigs, and poultry as well on the side. So that concludes the NB-GIP study that we had done. So now I'll progress on to the Charles University researchers and the work that they've been doing. Just before I do, I thought I'd have a brief recap on our knowledge about crude protein or protein and the way it is metabolized within livestock, ruminant livestock. So you've got crude protein, and that can be roughly divided into rumen degradable protein, non-protein nitrogen, and rumen undegradable protein. So we know that rumen degradable protein and non-protein nitrogen will get metabolized within the confines of the rumen, and rumen undegradable protein just sails straight through the rumen and gets metabolized later in the intestine. For an efficient process, we know when we're pushing production limits that our rumen degradable protein, if that is too high, then that leads to inefficiencies and loss of production. But if we have a high enough level of rumen undegradable protein, then we can essentially get an extra, I guess, wave of production from the intestine. Now, using standard NRC 2001 methods, we've estimated that the rumen undegradable protein in canola meal is about 20 to 40%. With a fair bit of research and some revised methodology, we now know that was underestimated, and it's closer to 40 to 60% rumen undegradable protein. The reason why that is because some of the soluble protein that we thought was metabolized into microbial protein within the rumen actually passes through the rumen into the intestine and is metabolized in the lower intestine. Now, if we look at the work that Rebecca Heim has done, she's completed a study of 9 canola meals from 8 different producers in Australia, and they're representing cold press, expeller, and solvent extractions. Her samples were analysed for protein fractions using NRC 2001 methods and also rumen degradability using a method from Krishnamurthy using streptomyces. So what she found was that cold press meal had lower soluble protein and rumen undegradable protein than both expeller and solvent extracted meal. All the protein fraction values generally agreed with overseas values, except on a whole, except for the non-protein nitrogen, which was lower in Australia than overseas values. Rumen undergradable protein had a larger range, 19 to 70%, which is quite enormous. And that was greater than both soybean meal and cottonseed meal, which were both studied during her program. So this is a summary of the results of her study in a table. And you can see from that the soluble proteins for the cold press meals were far higher, and the rumen undergradable protein levels were a lot lower. This backs up work that we've done at the Department of Primary Industries laboratory as well. We've seen a large range of soluble protein values and a fair range of rumen undergradable protein values as well. You can see that we use more traditional NRC 2001 methodology at the moment, which underestimates some of that rumen undergradable protein. Now, if I move on to the work of Miss Emma Lynch, she's a PhD candidate currently studying at Charles Sturt University, and she's looking into a study looking at optimum supplementation levels of canola meal that have been grass-fed, and looking at those gap periods of summer and autumn periods. And she's looked at a combination of pen feeding and animal house studies undertaken to look at the effectiveness of varying inclusion levels of canola meal as a grass-fed supplement. So just as a bit of a backstop to that, about 2/3 of production of livestock in Australia is from grass-fed industries. So within those, managing variability in pasture quality and maintaining consistent supply throughout the year is really challenging for certified grass-fed markets. So they look towards energy and protein supplementation within their requirements and to meet their requirements and production demands. And in recent years, canola meal has become more readily available in southeastern Australia. So there's a real opportunity there for canola meal there. The key findings of her studies were that digestibility and protein degradability increased with increasing levels of canola meal. She supplemented all the way up to 47% and had no negative effects in vivo digestibility. On top of that, the dry matter, crude protein and fibre degradation over time increased with increasing levels of canola meal. And also rumen pH, ammonia and VFA concentration significantly increased with varying inclusion levels of canola meal, and there was no negative effects up to 43% canola meal in the diet, which is quite substantial. That's solvent extracted canola meal as well. So just to recap on the opportunities for canola meal in Australia, there's opportunity to investigate genetic potential to increase protein content of the meal. There's looking at increased use in domestic livestock industries to fill feed gaps. That's a real opportunity. Value-adding products to support protein consumption for human nutrition. There's an extra one here as well, looking at methane reduction. Canola meal and canola oil have both been shown to reduce methane output in cattle of up to 10 to 30%. If it's too high, that it can influence fibre digestibility, so more work has to be done in this area. And Meat and Livestock Australia, which is Australia's peak body for livestock, has committed to be carbon neutral by 2030. So there'll be a lot more work done in this area in Australia. All right, and I'd just like to add some acknowledgements as well. So this work's supported by New South Wales Department of Primary Industries, the Grains and Research Development Corporation, Charles Sturt University, and Meat and Livestock Australia. Thank you very much.